Hi, everybody. Thank you for coming to my talk today. My name is Alan Zhu. I'm a fourth-year PhD student in the Tron-Hay Lab at the University of Chicago. Today, I'd like to talk to you about a study of the M6A RNA demethylase ALKBH5 and its role in acute myeloid leukemia. But first, some background information. What exactly is M6A methylation? M6A is the most abundant RNA modification on eukaryotic messenger RNA, occurring on 1 out of about every 2,000 adenosine bases. It's basically adenosine with this additional methyl group installed on it. RNA acquires M6A through a writer complex that consists of metal 3 and metal 14. It is erased by either FTO or ALKBH5. M6A can play big roles on RNA metabolism, and this occurs through M6A reader proteins that bind to M6A and then exert effects on the target transcript. For example, YTHDF2 promotes RNA degradation, whereas DF1 and DF3 promote translation. There are a few other readers that have other effects, such as nuclear processing or splicing. With these various effects on RNA metabolism, you can imagine that M6A plays a big role in cell biology. And so many studies have shown the effects of that. For example, in mice and zebrafish, M6A plays a big role in embryonic stem cell differentiation. It also affects pancreatic beta cell biology, T cell homeostasis, many cancers, as well as neuronal development. And here's some literature in case you're curious and want to explore more. A quick refresher on AML. It's a fatal hematopoietic cancer. And what happens is that myeloid progenitor cells are blocked in differentiation, thereby leading to clonal expansion and uncontrolled growth. And even with current available treatments, survival is less than 30% over 5 years. One root cause of that is that there are leukemic stem cells that have these unlimited self-renewal and repopulating potential. So even if there's treatment, as long as they exist, they can then lead to relapse. As a result, we need a better understanding of AML in order to create more novel therapeutic approaches. So that's where we come in, where we ask the question, what role ALKBH5 plays in leukemia? The reason we were interested in this question is that it was previously shown that FTO plays a role oncogenically in leukemiogenesis. Furthermore, other studies have shown that metal 3 and 14 can promote AML. But a previous study suggested, based on a TCGA cohort analysis, that ALKBH5 is deleted heavily in AML patients and is TP53 dependent. So we wanted to clarify that and then ask what role ALKBH5 plays in leukemia and get to the underlying mechanism as well. So we first did some analyses in TCGA cohorts and found that based on the deep deletion rate, the rate is actually low. The alteration frequency is less than or equal to 1%, which was lower than previously reported. When we looked at primary AML patients, even upon different types of leukemia, there was greater ALKBH5 expression compared to normal controls. And in TP53 wild type or mutant cell lines, you have greater ALKBH5 expression across the board. Finally, if you look at Kaplan-Meier survival curves, ALKBH5 expression is positively correlated with worse prognosis, thus motivating a study into whether ALKBH5 is necessary for the growth of AML. When we knock down ALKBH5 in a couple different cell lines, we see drastically reduced cell proliferation. Accompanying that, we also see that ALKBH5 knockdown increases apoptosis, as shown by this flow cytometry. And by reducing the M6A eraser, you have increased M6A methylation. Now, we then wanted to ask whether ALKBH5 could affect cell immortalization in leukemiogenesis. In order to look into that, we needed some in vivo models, so we used CRISPR in order to delete ALKBH5, creating hetero and homozygous models. And then we took those mice and made them donors, and then transformed their stem and progenitor lineage negative cells using MLL-AF9, which transforms these hematopoietic stem cells into inducing AML. We can then do colony formation assays or assays to assess leukemiogenesis with recipient mice. So then we asked the question whether cell immortalization is affected. With colony formation assays, you can see that if you remove ALKBH5, cell immortalization is basically inhibited. Furthermore, if you look at bone marrow transplants in recipient mice, removing ALKBH5 delays leukemia onset based on these survival curves. And finally, if you look at spleen weight as well as the immature blast cell population, you can see that depletion of ALK5 strongly decreases the percentage of immature blasts and weight of the spleen. We next wanted to ask whether ALKBH5 was not important for just starting leukemia, but also maintaining and repopulating it. So in order to do that, we took mice that already had primary AML and then knocked down ALKBH5 or had a negative control and put them in recipient mice. And as you can see, with knockdown of ALKBH5, you have impaired progression of MA9-induced AML in the recipient mice. And furthermore, when you did colony formation assays or xenografts of these cell lines, you still see that there's decreased colony number and size as well as increased survival, suggesting that ALKBH5 is important for maintaining AML. Going into the root cause of leukemia, we then looked at leukemic stem cells. In order to look at that, we transformed mouse bone marrow cells with a YFP positive marking MLLAF9 lentivirus and selected for the LGMP population based on cell sorting. And the LGMP population basically contains these stem cells. When we look at those cells, we can see that stem cells are in lower frequency when you knock out ALKBH5 and that there's increased apoptosis as well. In order to see whether this plays a role in repopulation capacity, we then did an in vivo secondary bone marrow transplant assay. And with knockout of ALKBH5, you had far greater survival and far lower white blood cell count, suggesting that ALKBH5 is important, and the knockout inhibits stem cell repopulation capacity. Now, I don't have time to go into all other results, so I'll just list a few of those things here. For example, leukemic stem cells express ALKBH5 at a much higher level than bulk AML cells, and this is not the case with normal hematopoietic stem cells. Furthermore, depletion of ALKBH5 doesn't affect normal hematopoiesis. And while these data showed knockdown, we also did forced overexpression and found that only wild type but not mutant ALKBH5 promotes cell growth. So if you only remember one thing from the first part of the talk, it should be that ALKBH5 basically induces a worse phenotype in AML. But the question is, how exactly does ALKBH5 mediate such an effect? And that's what we'll answer in part 2 of this talk. So we know that ALKBH5 targets certain transcripts, demethylates them, and that this will probably change the overall transcript or protein expression or abundance. So we were able to query that by using different high throughput sequencing approaches. And here's a review of how they work. You might be familiar with RNA-seq already. You take mRNA from your samples, fragment them, create cDNA, and then ligate these adapter barcodes onto them in order to create a library that can then be sequenced and analyzed. We also did RIP-seq, which is similar to RNA-seq, except you first pull down the ALKBH5 proteins and then take the RNAs that they're attached to find what RNAs are enriched. Finally, we did M6A-seq, similar to RIP-seq. You pull down M6A, and then you create a library of the M6A methylated RNA transcripts. These 3 different approaches tell you different pieces of information. With RNA-seq, you can get differentially expressed genes. With RIP-seq, you can get ALKBH5 binding target transcripts. And with M6A-seq, you get differentially methylated transcripts between your control and your treated samples. So we then did the sequencing, and starting with RNA-seq, I'll just give a summary. We found 6 and 1200 up and down regulated genes after knocking down ALK5. Now, in order to summarize that, we looked at gene set enrichment analysis profiles, and we found that with ALKBH5 knockdown, you have suppression of E2F targets, MYC targets, and cell cycle checkpoints, as well as activation of apoptotic and P53 pathways, which matches up with what we found in part 1. To summarize RIP and M6A-seq data, we found almost 1,400 and 500 differently methylated targets, and a lot of the same pathways are similarly enriched. But what we want to find is a high confidence target of ALKBH5 and AML that might be responsible for the phenotype. So in order to do that, we wanted to find genes that satisfied these 3 conditions. Are they differentially expressed upon ALK5 knockdown? Are they bound by ALKBH5, which RIP-seq will tell us? And after an ALK5 knockdown, do those transcripts have increased M6A methylation? When you find genes that satisfy all 3 of those conditions, you actually get just about 18 potential targets, 12 that are down and 6 that are upregulated upon ALKBH5 knockdown. We validated these findings with qPCR, and we decided to focus on TAC3. The reason is that when we look at the TCGA AML cohorts, TAC3 high expression leads to an adverse prognostic impact in AML, but we didn't see the same effect in all the other targets that we found from sequencing. When we look at gene tracks, this validates what we previously found, which is that upon ALK5 knockdown, you have less expression of the transcript, and you have greater methylation of the TAC3 gene. We also validated this with cell lines, using different cell lines as well as a primary patient. If you knock down ALKBH5, it concomitantly leads to a lower TAC3 expression. So then the question is, how exactly does ALKBH5 post-transcriptionally target or post-transcriptionally regulate its target TAC3? It could be through transcript stability, or it could be through translation, just to name a couple things. So we first looked at mRNA of TAC3, and with knockdown of ALK5, you get less mRNA transcript abundance. And this is because you also have less mRNA transcript lifetime upon ALK5 knockdown, suggesting that ALKBH5 controls the transcript stability of TAC3. When we looked at translation through polysome profiling, we can see that the polysome fractions are not significantly affected, suggesting that ALKBH5 regulates target transcript expression through RNA stability rather than translation. So then what exactly does TAC3 do then that affects myeloid leukemia? Well, previously it was shown that TAC3 could control apoptosis through P21, and that less TAC3 also leads to less MYC in hepatocellular carcinoma. So we looked at those two proteins in acute myeloid leukemia, and upon ALK5 knockdown, you see less MYC as well as increased P21, which matches these previously observed trends. We also found that even if you knock down ALKBH5 and see less proliferation, you can rescue it with forced expression of TAC3. Furthermore, this Western blot shows you that if you knock down ALKBH5, if you express TAC3, you reverse the effects on MYC and P21. And while I don't have time to go into this, TAC3 depletion also phenocopies ALKBH5 deficiency. So overall, this leads to a general model in which ALKBH5 demethylates M6A from the TAC3 mRNA transcript. This leads to greater MYC as well as less P21, leading to greater self-renewal and less apoptosis, thereby leading to the more deleterious effects we see in AML. There's plenty more results that I didn't have time to go into today that you can check out in a paper we published in Cell Stem Cell earlier this year in July. I'd like to thank my advisor, Chuan He, as well as my collaborators, Professor Chen and Chao Shen from the City of Hope Cancer Treatment and Research Center. And I want to thank all these lab members in my lab for making my MD PhD experience so wonderful. And I want to thank you all for coming to my talk, and I'll be happy to take any questions.